So far on the line here, we have Jim Adams and Rosie Krajmalnik-Brown, obviously Joan Fallon. Gahan Pandina is here still. Miraculously, it looks like Véronique Crutel is still online from France. So she's a trooper. Bob Hendren is on. And so please feel free to, maybe I'll open the floor up to the panelists to ask any questions of anyone else who's on the phone. I see Kevin Saunders is back on as well. So any questions? I'm hoping that people will write in questions. I think maybe one thing to get things started here is we've seen now with the Curemark work going toward irritability, the Axial work going toward irritability. We saw in the Servier talk focus on just overall autism core symptoms using the CARs, and several others mentioned irritability as well. The Roche folks have been mostly focused on adaptive behavior, in particular social communication. So it strikes me that, to your point, Joan, earlier, that there's not going to be a one-size-fits-all here, right? And so at some point, look, if we're all fortunate enough to get these therapies approved, let's assume that for a moment, the next step would then be combining these in some way to get the best outcome. And in fact, someone asked the question earlier about whether you could even add some of these therapies to someone who's had a microbiota transfer. So maybe with that kind of preamble, I'll pick on Jim Adams first and say, if there are things that, I mean, you obviously have this sort of wholesale replacement of the microbiome, which one could say has the potential to make many of these other things kind of irrelevant in a way. I mean, that would be, you know, if you could actually change everything. Do you see a likely scenario where the MTT therapies would then be supplemented or combined with people who are on other therapies? Would you look at the MTT as a last resort or a first call type therapy? Maybe speak about that a little bit.
Sure, and Rosie, feel free to chip in too. My thinking is that MTT has a lot of benefit, that we're restoring what's hopefully a fairly normal microbiome, and the microbiome plays many roles in the body. It helps with producing certain key vitamins, with water balance, with digesting food. It helps with many things. But it doesn't, I don't know that it would be directly affecting chymotrypsin levels, so I'd be curious about that. We have done a number of studies, and have been quite a few studies of amino acids, and unfortunately many of the studies of amino acid levels haven't been very well done. But when you do them carefully with fasting, with controlled reference ranges, etc., then it seems like the differences are somewhat small. So I'm curious, a question for Joan I might ask is for, did you measure amino acid levels in the Curemark studies, and did you see changes in those?
We did not do that. We did not do it. And I think one of the things that we were sensitive to initially was whether we would have a blood draw or not in the study, right? Because that, of course, changes the character of the study and the measure of irritability at a visit. And that's kind of where we decided not to do that. I mean, I think there's been a lot of work done on the amino acids. We know that there are some deficiencies, et cetera. But with this sort of very low pathological level of chymotrypsin, again, you don't have a lot of redundancy. So we use that as sort of our base, and hope to look at that more in the older children. We looked in our first study at changes in food intake in a very sophisticated way. and found that there was a significant difference in food intake, the kids on the drug versus the kids not over a 12-week period. So there's some things going on. And I really do feel they all have an interplay. When we look at certain pathogens, in our study, we do stool pathogens, some of them, not many of them, certainly not looking at a full microbiota, but I think that there's definitely effects here there's an interplay between the microbiome, the enzyme, all the pieces of the microbiome that go together. Because it all happens in one place. Look, the amino acid uptake pathway sits right next to, the receptor for the SARS-CoV-2, right there. It's right next to each other, right, in the same place. So there's a lot of interplay here. And there's more receptors for COVID in the gut than there are in the brain, in the heart, or other places. So there's a whole other issue there that goes on, I think, that we're just beginning to understand a little bit of.
Yeah, and I think the whole role of diet has a huge effect on your microbiome as well. And that's one thing that we'd like to measure in our next study, is to see more carefully how diets change, especially because many children with autism have such self-restricted diets. Rosa, would you like to comment more on that?
Yeah, I also want to add that, ideally, if I could have my wish of how I would like to do this therapy, it would be with a cocktail of microbes, not with microbes that we can grow in the lab, not with microbes that come from, donors from fecal samples. So ideally, with the studies that we're doing, we can learn more about which are the microbes that are important. And I don't think it's one. I think it's just like we're talking, there's going to be more than one. But if we can find a group of microbes that are important, that if we use those microbes or change those microbes, I think that would be, in my opinion, a more target and better therapy that using fecal samples from a donor, which we might have gotten lucky and that's why it worked. Maybe we had the right donor and that's why it worked, but we don't know. We don't have a larger sample yet to know. And I don't think that MTT fixes everything, and I don't think that it's a magic wand. So I think we need to learn more about it. And you are what you eat, because what you eat crafts your microbiome. So that together with what you eat is important. I found it really interesting that I didn't know about this protease thing with kids with autism, because that ties really nicely with all the metabolites that we find that are the product of amino acid metabolism, because you have a higher amount of microbial amino acid metabolism in the gut, which makes sense because you're getting more proteins there.
Right. It all goes together. I mean, there's a very big overlap, and I mean, think about 10 or 15 years ago, we wouldn't be having this discussion, right? And now we're having it in a robust way, which I think is really important.
Mm-hmm.
Right. I think there are also several studies that show low levels of lactase in children with autism, where they actually measure it during colonoscopy. And so 3 different large studies have shown that, and that's probably why many children seem to benefit, part of the reason why many children seem to benefit from a lactose-free or casein-free diet.
Right. And casein is a protein, right? So
yep.
If you can't digest it, you're going to have a problem with it. That's just sort of how we've looked at it.
Yeah, so dairy has multiple problems, also the effect on the cerebral folate transporter, developing antibodies to that. And we heard a number of presentations earlier in the sessions about that.
So we've got a couple of questions in. This question is for all panelists and is sort of up at a sort of very strategic level. At a systems industry level, what bottlenecks do you see that, when addressed, will accelerate the development of effective therapeutics for autism over the short and medium term? Maybe I'll first direct that question to Kevin Saunders and then to Gahan Pandina, if that's okay, and start the discussion.
Sure. So the first thing I guess that comes to mind are And I think I mentioned this earlier, reliable endpoints. I still feel like, I mean, for some areas like irritability, maybe they're different, but for the other core symptoms, I feel like they're still very lacking. So I feel like, as endpoints evolve, as we can evolve with maybe even digital endpoints to get more objective measures, I think that will move the field forward tremendously.
Yeah, and I would say, some of these larger consortia efforts in just trying to establish good measurement properties, good approaches, having more sites with expertise in doing research. I find that there are really excellent clinical centers of autism that do autism work, but don't have the infrastructure for research. And then, although there are some research centers that focus on research, they get swamped with lots of protocols and then their own research. So it's I think creating there's now a much better environment for optimizing that research. It's also would mention that, given that a lot of the focus has been on the US, having much more expertise and a larger group of researchers available in Europe, particularly for global registration programs and planning for good trials, is really important. And so the tools and the technologies will help us. Some of the basic science research will help us. Some of the aggregation of data will also help to just begin to give us a better structure for doing all of those programs.
I'd like to comment on the issue of assessments. I think one of the challenges, the big challenge we have, is that with the clinical assessments, the clinicians have a lot of experience, so they can calibrate the severity of a child reasonably well, but they only see a child a very limited amount of the time. So even with, say, a formal ADOS type evaluation, they're only seeing the child that day. And kids with autism can have very good and very bad days. And so they may be able to assess that child's severity very well, but not be able to assess changes that well because they only see the child for that snippet of time. Parents aren't able to calibrate very well, but they know their child very well. So what we found is better to do with parents is to ask them about the change in the symptoms. So rather than having them say, here's my absolute score on this day, and here's my absolute score this day on a scale of 1 to 10, they can't do that very well. But if you ask them how much change has occurred during a study, we found that's been much more reliable than having them fill out a form like the ABC or SRS, where they have trouble calibrating. Well, do I give them a 2 or a 3 today? So I think using assessments for parents that ask them about the change in their child over the course of time has the advantage that the parent or a teacher sees a child many, many times, as opposed to a clinical evaluator just seeing them for a snippet of time at beginning and end of a study. Unless you have someone like a behavioral therapist who's with the child many hours a week. That would be, I think, perhaps the best way to do assessments.
We're excited to look at the data, the long-term data that we have for the kids who've been on this treatment for such a long time, and now they're home with COVID. I mean, they're in home during this COVID time, and to look at those assessments and what they look like now. It'll be very interesting to for us to take a look at that. So when we have enough of it, we will really look at it. But I suspect it will be different than sort of the trajectory that they've been on all this time. And whether that's a function of them or function of their parents rating them, I don't know. It's a very different purview they have now that the kids are home all the time.
I think it's very commendable you're doing those long-term assessments, and I think that's very, very helpful. I think a limitation of many studies, like with Risperdal, is if you do a short-term study, you don't see some of the long-term adverse effects like weight gain. So I think it's great you're doing these long-term studies. It gives us a lot more confidence in the safety.
We have, large ends out 108 weeks. I mean, we have, some significant data out there.
That's great.
I don't know if Bob Hendren is still on or not, but it'd be great to get his take on some of these assessments as well, and where he thinks the problems may be or the improvements may come from.
I don't know that I'm prepared to answer all of that.
Sorry, I put you on the spot. Sorry.
Interesting that what people have brought up are the different mechanisms. So if we think about AB 2004, we're talking about this little envelope that surrounds the neuroactive particle and helps it pass through the gut without creating irritation. I think where some people got started was with probiotics, and I think Jim mentioned a study that he's doing with tailor-made probiotics for people. Then there's Joan's using the a digestive enzyme that helps break down these large proteins so that they, at least my limited understanding, pass through the gut in a way that doesn't irritate the gut lining to create so much of this opening of the juncture that people, or we used to laugh about when people called a leaky gut, but now we somehow say, yeah, I guess I understand how that could happen, that these things are leaking into the system and they're causing problems. How do we help with that? I think they're all different mechanisms, and the idea that people are bringing up is to say people might all be different, and we need to figure out subgroups of people that would respond and not respond to one approach or another approach. And that's kind of the big dilemma in all of autism. It seems that we keep struggling with subgroups that we hope we can do better with metabolomics and subclassifying, but we haven't gotten there yet.
Thank you. Véronique, you look like you're still with us, you're still awake, so maybe you can give us your opinion on this as well.
Yes, I just want to say that I fully agree with my pharma colleagues who just said, of course, that we need clear outcome measures, we need very good sites, but I think we need as well reliable biomarkers that would also be really helpful in the development, of course. That's a crucial point as well.
Thank you.
I guess the other quick comment I'd make is how interesting it is that we're talking about this now in ways that we wouldn't have. I recall maybe 10 years ago, I reviewed a grant from somebody that was wanting to use the cannabidiol system to treat autism. And I kind of chuckled and I thought, oh my God, everybody's promoting marijuana. If you just smoke marijuana, you're going to feel a whole lot better. And then I see this serious science coming along, and Gahan's presentation that talks about what's involved, or the way we used to think about the gut. And even at the beginning, when Joan got started in this for Curemark a number of years ago, I think there were lots of skeptics. And now, or for Kevin and Roche thinking about using something like the vasopressin system that might play some role, it's like we're moving into a new era that we wouldn't have taken seriously a while ago. And I don't think we're there yet, but I hope that it's moving us in a way that can get to the complexity of autism.
Well said, thank you, Bob. That's a great summary of, I think, where we are, and we all wish we were at that next level already, but I think we just have to keep doing the hard work. Another question, and to that end, what would be the top 3 suggestions for tactical and strategic ways that a nonprofit like the BRAIN Foundation could undertake to facilitate development of effective therapeutics? Gahan, maybe I'll get you to start, because you had talked about the need for some of these centers, academic centers and institutions that are maybe best equipped, but then they get inundated because they're with protocols because they're best equipped. And how do you create that balance between doing the fundamental research and moving therapeutics through development?
It's a great question. I think we've faced this a lot in the last 5 years. especially with many single gene disorder foundations springing up. And one of the things that I've been so impressed with in those efforts is, first of all, having passionate people committed to seeing it through. So they're not putting up a website and saying, we're not really going to follow through with this. This is something I was interested in because I have a personal investment, but we'll let it sort of pass. They've really started to focus. The other thing that they've started to do is to help gather data. So they've created through different mechanisms, including some orphan disease structures within NICHD and NIMH, the ability to begin to gather case information and longitudinal data that informs the field, and also can funnel data into basic science research, can help get access to populations, and also create contact points. Sometimes companies have a difficult time to talk with patient groups and create meaningful outcomes. They're really focused on what their clinical endpoints need to be and how they're going to satisfy the regulatory authorities. And regulatory authorities are looking to the field, and the field is trying to generate these outcomes, but it's really difficult work to do. So I think one thing that nonprofit organizations can do is to help the field organize and to begin to use those basic structures to provide better data and better reference information for us. And also networks that allow people to get access to research. Many patients and families don't know who their local experts are. And now it doesn't so much matter as much as it used to, but it matters that they get connected to those resources, not just to the researchers, but then to resources. So I think as we improve care, as we improve connectedness, and as we improve those tools, that's to me the environment that the nonprofit can help with. It can also help a bit with fundraising, but as I've said, if you can raise $10,000 to do a study, given what studies cost, that might not help as much as it might help a new researcher to follow out a new experimental idea. I think gone are the days of a big nonprofit funding large multi-center randomized placebo-controlled trials for the sake of generating new data in one area. I think they're best served doing it another way. But I would welcome opinions from the panel as well.
Gahan, what you bring up is really important in terms of how you get information disseminated about some of these conditions. I remember when I was in practice, I had a young man who had Cockayne syndrome, which is a progeria-like disease. And, he was not mobile. He died at the age of 5 after looking like a very old man. And the family decided they were going to fund some research. So there's a researcher at Harvard who's doing the leading work in this area. And he and I sat down and he said, what was the most profound thing about that child? I said, he never got sick. Here's a non-mobile child who never had an ear infection. He just never got sick. And the research said to me, none of them get sick. So here he's aggregating data, realizing that these children, when they should get sick, just normal childhood illnesses, they don't get them. So having that kind of information can lead to a lot of discovery. But if we don't have that communication, it's not going to happen.
So what I'm hearing here is that the nonprofit side of things, the foundations that focus on whether it's particular populations or something different, it's really on the epidemiology side of things, right? So it's not just, so it's phenotypic profiling, genetic profiling, metabolomic profiling, microbiome profiling, whatever other omic thing you want to throw in there, but it's really about assembling that data and making it available to researchers, right? Because I think what you described, Joan, is a very specific case where that person, that researcher at Harvard, probably because there's so few of these people, knew most of them, right? Yeah. And then you take something like autism where it's so prevalent. How do we translate that concept to a very large population that's very diverse, I guess?
I think, autism is many things, as we know. And so with these many groups that we've already begun to identify, understanding their needs, engaging them, having them engage with these larger consortia and research efforts, having them help to drive the research agenda with important questions to them, to families, and important observations, and having that information, not just that they have to aggregate and find ways to do that themselves, but participating and working with their local research groups and thinking about what's important to them in their clinical care. Those are also important endpoints. And I would like to see, we just held a really nice program on orphan disease at the ISCTM a bit ago in the fall. And one of the things that came up is how do we get participation from patient and parents and advocacy groups in early research design and mechanisms to do that. So it's not just necessarily raising big funds to get these big epidemiologic studies funded, or gathering all the information themselves, but figuring out ways that they connect to the various research entities, and how they help connect clinicians into those entities so that we can facilitate better research.
Very important distinction. Thank you. That's really, really good. So there's a couple of questions here from the participants. One on the microbiome. This one I'll just read verbatim. We keep hearing that the ASD microbiome is different than typical. However, lab tests like 16S RNA or whole genome give a wealth of data that is hard to interpret even by some doctors. What needs to be done to get a reliable and easy to understand test? Is this available, more standard in research settings? I guess that's a complex question. So I'll turn it over to Dr. Rosie for that one.
I think it's a great question because you get a collection of sequences. You don't always know what it is, right? And I have had people ask me about their microbiome and show me reports like that. And I'm not a doctor, so I can't provide a lot of or any medical advice. And there are certain microbes that we think we know what they do, but it's difficult. I don't feel that the field is there where we can take a microbiome report and say, this is a problem, this is what we need to change, and this is how you change it. I don't think we're there. That would be great. And that's where we're heading, and that's where we want to be, but I don't think we're there. And so that's one of the challenges. I don't know if Jim wants to add or.
Yeah, I think the best way to think of it is the microbiome is a very, very complex collaboration of hundreds to thousands of different species. A good analogy, I think, is a village. If you have a village, you want to have a doctor there, you want to have a grocer, you want to have an attorney, a teacher, etc. But if you have 100 teachers or you have 100 attorneys, that's not a very good balance. So what we want to do is have a balanced microbiome. And diversity is one simple measure that I think most people agree a higher diversity is generally viewed as more healthful. And that's what we found in our studies, but not all studies have found that. There's a lot of disagreement between different studies of the microbiome and autism. There are some labs out there that I think can give some good insight. For example, with Sun Genomics, they do give a nice summary trying to interpret these are generally beneficial, these are generally harmful bacteria. But again, it's a question of what's the balance. But also they have a very simple score, 0 to 100. Where do we think your microbiome quality is? 30, not very good. 70, that's pretty good. So if you want something very simple, that's one approach to take, but each lab has their own way to report their results. But the bottom line is we still have a lot to learn about what is optimal.
So Jim, I'm going to add with your analogy, which I really like about having, one lawyer, one doctor, one is not enough, right? And so we need functional redundancy, because if the one doctor of the village gets sick, then who's going to take care of the problem? So yeah, there's that balance where you need the functional redundancy, which is why you need high diversity. But the other thing is that microbes are not always good or bad, and it depends on the environment they are at, how they behave. So for example, I show the sulfitobacterium, which many people will tell you, this is a bad guy. It produces hydrogen sulfide. Hydrogen sulfide will actually damage the lining of the intestine. It has been actually correlated with, I think, IBD. But in our case, it looks like the sulfitobacterium might actually be doing something good. It's a challenging question, I think, because of that.
Right. So if you only had Lactobacillus acidophilus in your gut, that would not be very healthful. And so again, what we want to do is try to establish balance. So I think the Sun Genomics approach in general, at first, let's see where your gut is at, and then let's try to use an appropriate combination based on that. I think it's that sort of thinking, and we have a long ways to go in improving algorithms, but I think it's that sort of approach of not just saying, here's one probiotic to help everyone. It's not what we need. What we need is to try to restore balance. And FMT is one way to do that, but that's pretty invasive. And so as Rosie says, it would be great if years from now we can figure out what's a good mix that we can grow.
And you also need a holistic approach that takes into consideration also the host metabolism, that links the host metabolism with the microbiome.
Great, thank you. There's a suggestion from, quote, outsider here, and this is not from me, I swear. So it's going to sound loaded, but it's not. Here's a suggestion: connect companies who need collaboration to move forward. You have the network that small companies may not have. And I don't know if it must be the large companies out there or the large institutions. Helping small companies making the right connections can be more important than standard funding, and it can create a much better use of your money. So that's just a suggestion or a comment from somebody who's in the audience saying, and I think it probably is a permutation on what Gahan was saying earlier, right? Which is find a way to connect people and not just fund individual research. Because anytime you fund a project, the project has specific goals. The researchers are motivated to deliver on those specific goals, when that specific goal could be successful outcome in a clinical trial, which is usually designed to be successful with a certain endpoint. And so by definition, you've taken these sort of global connections you could make or global sets of data you could pull together, and you narrow them down into very specific questions. And that may answer that specific question, but it doesn't help anyone else more broadly, I think, or at least it helps them less than it could otherwise. So.
Stewart, I might try to answer that a little bit differently. I like a quote from Sid Baker, who says, if you're sitting on 10 tacks, taking out just one of them only helps so much. So chymotrypsin, low chymotrypsin may be one tack that we want to remove. Abnormal gut bacteria, another tack we want to remove. So different children are sitting on different tacks, if you will. I think our goal is to figure out what are effective treatments for a given problem, and then have a series of treatments that hopefully is chosen, tailored for that individual, based hopefully on biomarkers or something else. So they don't have to try each one randomly, but can hopefully then say, oh, you have low blood sugar, you need some insulin. Oh, you have low chymotrypsin, well, let's give you this digestive enzyme.
Right. But it's like, to me, I look at like depression. There's like a lot of drugs for depression, right? And some of them work for some people, and some of them work for other people. And some people use polypharmacy. It's like, there's a lot of different combinations of things as people are different, right? So we never looked at what we were doing as the answer. It's a treatment for some people.
Right. Just depression, like autism, is a symptom, and many things can cause that symptom. I think the approach we should be trying to take is, what are some fundamental problems in autism, be it low chymotrypsin, being it abnormal molecules from the gut, and let's come up with treatments for each one of those. But then I think we really need to try to work with families and physicians in the next step of how to integrate those treatments in a meaningful way.
All right. So I have no more questions from the audience at this point. I wonder if anyone has any final comments or questions for anyone. That'd be great to hear them now.
Hey, Stewart, it's David. I just wanted to thank you for a great job moderating today.
Well, thank you. It was a lot of fun. I learned a lot, actually, so that was good. I guess on that note, I would like to take the opportunity to thank Pramila and her team for putting this on, and for everyone who was able to join and present. A terrific day or night in the case of some people over in the other side of the Atlantic. I really hope that this is the start of something. I feel like we need to do this more because I find them very motivating, I guess, and makes it, it's great to see so many people trying to address the same problem in different ways, or different aspects of the same problem in different ways. And everyone's just so willing to cooperate and really just wants to make it better for the families and the kids. So we'll get there. And I think, you know, if this is any indication of where the field is headed, I'm really, really excited. So thank you, everyone.